So we're going to wrap things up with hemoglobin and myoglobin. And before we do that, we're going to talk about something called cooperativity. So the idea here is pretty simple. I know that this cartoon reaction maybe doesn't look so simple, but let me walk you through it. So in the first step, so it's very top, we have two different protein chains. We have a left side and a right side. And what we're going to do is first we're going to look and notice that the chains are colored. Blue means that it's a stable or rigid conformation of that protein. So it doesn't move around a lot. There's not a lot of flexibility. It looks the way that it looks. It doesn't change very much. On the opposite end of the spectrum, we have red, which is totally unstable. It's very flexible. If you were to look at this tail in solution or look at the structure of it, you would see that the red part flops around a lot. And then in the middle, you have this green linker section. And the green section has a little bit of flexibility and it has a little bit of rigidity. So it can move around a little bit, but not too much. And we notice too that the green section butts up against the green section for the neighboring protein that it's connected to. So if we walk through our first step here, our first binding event, we're going to look at the chain on the right side, and we're going to add a ligand to the binding site. And what we see is the blue part stayed blue, which means that it's immobile. The red part turned green, which means that it still has a little bit of flexibility to it, but it definitely has some defined structural regions that don't move. And then our linker region in the middle turned blue, which again means that it's well structured. It doesn't flop around at all. It just stays exactly where it is. Now, the interesting thing here is that making this backbone region or this connector region rigid like that actually transmitted information into the neighboring protein and made its backbone region, or sorry, made its flexible region pretty rigid and pretty structured. So then we go to our second binding event and we see the same thing happened, right? The binding site stays blue. The area that was red turns green, which means that it's got some order, but it still has a little bit of flexibility to it. And then we can go backwards and reverse, right? So this is cooperativity. Binding event on one binding site gets transmitted through structural perturbations into the neighboring binding site or the neighboring protein chain. And this has implications for us in myoglobin and hemoglobin. Well, sorry, implications for hemoglobin. Myoglobin is a monomer. It exists as a monomer. It doesn't interact with anything else, so there are no allosteric changes to speak of. And I just realized that the speech-to-text software did not pick up allosteric. There we go. That's what it should have been. Okay, so right, so we're talking about allosteric and cooperativity. So I don't really care too much about homotropic versus heterotropic. I'm not concerned about those words. The thing that I am interested in here is just the idea of an allosteric protein. Right, so like I said, when you have an allosteric interaction or an allosteric binding event, something happens in one part of the protein and it's felt on an opposite end of the protein or even in a different protein nearby simply because of a chain reaction of perturbations in the structure. Okay, so hemoglobin, right? We said that hemoglobin is a tetramer. That means it consists of 4 subunits, 4 monomers. And in our specific case, we have 2 alpha subunits and 2 beta subunits. Each one of those subunits looks very, very similar to myoglobin. It has nearly an identical structure. If you were trying to tell them apart, you really couldn't. If you were to look at the amino acid sequence between the two, then yes, you would see that there are differences in amino acid sequence, but not enough to give you differences in structure or differences in function. So we still have the same structure that we've been talking about, and we're going to wrap 4 of these hemoglobin subunits up into quaternary structure, and we're going to make one functional hemoglobin complex. So here we have alpha in gray. There's alpha 2 in the top left. Here's alpha 1 in the bottom right. Then we have in blue, we have beta 2, bottom left, beta 1, top right. And you can see that the alpha and beta monomers form this kind of crisscross structure. If you look at it in a particular way, you can see that there's an empty central part in the molecule. We can see the 4 individual heme groups sitting between, I believe it was helices E and F in each of the individual subunits. So beta 1, alpha 2, beta 2, and alpha 1. And one other thing that we should talk about is the area of interaction between each of the monomer subunits. So if we look in this vertical axis here, right in the middle, look at the region between alpha 2 and beta 1. We can see that there are really only probably 2 helices that overlap in this region. And if you look carefully, this is not something that you'll find out, but this is someone sitting down with a piece of software, looking at the structure carefully and counting distances. Distances that are less than, say, 4 angstroms, something like that. If you do that kind of analysis, what you'll find is that there are about 19 amino acids between alpha 2 and beta 1 that interact with each other. If you compare that, right, so let's come down here to the horizontal axis and go right through the center of the protein. So if we look at the interface between alpha 2 and beta 2, which is right here, so that interaction is actually much bigger than the alpha 2 beta 1 interaction area. And what we see here is that there are about 30 individual amino acids that help make that connection between the alpha 2 beta 2 subunit. So alpha 2 beta 2 and alpha 1 beta 1, those interactions are strong, 30 amino acids, and it's much bigger. The alpha 2 beta 1 and beta 2 alpha 1 interactions are much smaller, only about 19 amino acids. And that's what holds this molecule together. I should also say that the dominating intermolecular force here is the hydrophobic effect. Sure, there are some hydrogen bonds, there are some electrostatic interactions, but if you look at the interface between the 4 monomers, the hydrophobic effect is what dominates and what really, really holds this molecule together. Like I was saying, there are some hydrogen bonds and electrostatic interactions holding it together, so that's what we're seeing here, right? So just as an example, between beta 2, the top strand here, and alpha 1, this next strand, there is an interaction between this carboxyl group and this lysine. We can also see an interaction between the N-terminus of alpha-1 with the C-terminus of alpha-2. We have mirror image interactions between this aspartic acid in alpha-2 with the arginine in alpha-1. And then, like I said, a mirror image pair, the aspartic acid in alpha-1 to the arginine in alpha-2. So when we take all of this and add it together, what we end up with is a protein that has 4 binding sites. Each one is comprised of something very similar to myoglobin, something that is an all-alpha helical structure, something that has a heme group that contains an iron atom and is able to bind oxygen. So what we end up with is 2 states. So we have the T state, which is the tense state. And this is in the absence of oxygen. And what you can see is that there's this large gap in the middle. But if you look at the individual monomers, they're tense, right? All of the alpha helices for the beta-1 monomer, for example, are tightly drawn in and seem to be much closer to the heme group than in this other R state, this relaxed state. So the T state is without oxygen. And the T state is a little bit weird because it actually kind of prevents oxygen from moving in there and binding to the heme group. But the really cool thing, and this is where the allosteric or cooperativity part comes in, as soon as oxygen binds to one of the binding sites, those structural perturbations in that binding site get transmitted to the neighboring monomers. So, for example, if we were to bind oxygen to the heme group in beta-1, beta-1 would change to the R state, and as it opens up, it would impose or it would send those structural perturbations over to alpha-2 and alpha-1, which would also adapt the R state and make them more susceptible or more open to receiving an oxygen molecule. So as soon as oxygen binds to one site, the other 3 sites open up and become easier to access. And if you look at this conformational change, we can see that in the T state, the histidine is actually pulling the iron out of the heme group just a little bit, right? So the heme group is sitting a little bit above the plane of where that iron atom is. The iron atom is being pulled closer to the histidine, and that would make oxygen harder to interact with or harder to donate its lone pairs to the iron atom in the middle. In the R state, on the other hand, the iron atom gets pushed out and sits exactly halfway in the middle of the heme group, right? So there's equal amounts of iron on the inside next to histidine F8 and then on the outside where the oxygen would bind. So that makes it easier to access for an oxygen molecule. Now, we've spent a lot of time talking about different amino acids that are involved in this, and many of them are charged. So these histidines specifically, they have acid-base chemistry, which means that the binding potential or the binding curve for hemoglobin depends on the pH of your blood. So at a pH of 7.2, the carrying capacity or the occupation of oxygen in the binding sites becomes harder because there are extra protons around, and that means that those histidines get a little bit more hydrogen, or they see a little bit more hydrogen than they normally do. So the binding curve shifts down, or shifts away from what we would consider being easy to put an oxygen molecule in. At a pH of 7.6, on the other hand, this is more basic, right? So we're starting to pull more protons off of things. And I realize that this is a small change. It's only a pH change of 0.4, but it's enough that we're starting to pull protons off of those histidines, and it makes it easier for them to interact with the iron atom in the heme, and that gives us a more open binding site that's easier to put an oxygen molecule in. Okay, the very last thing that I wanted to talk about is just a simple but important mutation to hemoglobin, right? So I think many of us are familiar with sickle cell anemia and how terrible it is. It really is just a simple genetic mutation, specifically at glutamate 6. Right? So glutamate or glutamic acid is a charged residue at physiological pH. If you substitute that amino acid with a valine, it's roughly the same size, but instead of being a charged amino acid, it's now a hydrophobic amino acid. And this new valine side chain can actually make hemoglobin molecules that interact with each other in ways that they're not supposed to. So just to show you what I'm talking about, our old friend, the molecule of the month, and hemoglobin. So right here's the article about hemoglobin, and if we scroll down, well, hey, look at that cool animation between oxygenated and deoxygenated and the structural changes associated with it. And then here is that mutation that we were just talking about. So this is glutamate number 6, or glutamic acid number 6, and if it's changed to a valine, like I said, it becomes hydrophobic, and it allows hemoglobin to interact with itself in ways that it was not supposed to. So you end up with these long threads of hemoglobin molecules, and that inevitably causes what a normal red blood cell should look like, this kind of donut-looking thing, to mutate and change into this elongated, sharp, abnormally shaped sickle shape. Okay, so to wrap things up, right, we talked about how ligand binding is super important in a lot of different areas, and we focused on hemoglobin and myoglobin as our case study because those two molecules are the most studied and most understood of all, and they were the first two to have structures done. Right? We looked at how myoglobin stores oxygen. We looked at how hemoglobin transports that oxygen around, and the similarities between those two molecules.